Madison Parker, and I'm an agricultural education student at Abraham Baldwin Agricultural College. And today I want to talk to you a little bit about genetically modified organisms. With the population expected to have a steep increase by the year 2050, agriculturists are striving to find ways to increase a healthy yield in order to satisfy the need to feed the increasing population. Genetically modified organisms, or more often referred to as GMOs, have been a prime solution to problems such as resistance, environmental intolerance, and lack of desired yield. According to a study done by Consumer Reports National Research Center survey, more than 70% of Americans say that they do not want genetically modified organisms in their food. But do they really know what they are discriminating against? The marketing ploy that numerous packaging and production companies are using to bump up their prices is extremely misleading and allows consumers to infer for themselves that GMOs are unhealthy and even dangerous. So today I wanted to discuss what GMO crops really are and what the science behind it is, so we can discuss why they really are safe and beneficial organisms to us. While some consumers may picture GMO crops to look something like this is all they really are. So let's discuss the science behind it. So it's all about genomics. Synteny, to be specific. Synteny is the physical co-localization of genetic loci on the same chromosome of a chromosome within an individual or species. Biologists usually refer to synteny as the conservation of blocks of order within two sets of chromosomes that are being compared with each other. DNA sequences are like a map of genomes, that is. Imagine if this happened. You lived in a city your entire life, so you basically have the map of your hometown memorized. You then move to a new city only to find out that your new town is very similar because almost all cities are the same, which makes navigating your new map much easier. Well, that's how it works with genetic sequencing of plants. Many plants have significantly similar genetic maps, which makes it easier to engineer them. In fact, 70% of the world's foods share a syngenous relationship and are linked in a genetic map that looks somewhat like this. For instance, the 12 basic chromosomes of rice have synteny and align with the 10 main chromosomes of corn. This is important because corn completes C4 photosynthesis, which is much more efficient than the C3 photosynthesis, allowing scientists to try to isolate the desirable genes in corn and develop a C4 rice, which will produce a greater yield that is more tolerant to harsh weather. In addition, the knowledge of the entire sequence of rice allows scientists to predict the order of genes that are syngenous with wheat and barley, identify the shared tolerant genes in rice, and isolate those genes in wheat and barley, making them tolerant to problems in soil and diseases. A tool used to increase yield and tolerance is the marker-assisted selection. A molecular marker is a sample taken from within an organism. The use of a marker-assisted selection, or MAS, speeds up the traditional process of breeding with desirable traits by years. DNA samples from the organisms are obtained and extracted are mixed with the salt buffer so that an electrical field can be sent through the DNA. As the fragments move towards the positive pole of the field, the resistant and susceptible traits will become visible by the links of the DNA fragments, as you can see in this image. The DNA that shows positive for desirable traits is kept for breeding, allowing farmers to transplant this DNA to be used in the field or move on to micropropagate that desired organisms to produce crops that are resistant to disease and drought, for example. The technique shown in this image is micropropagation. Micropropagation is a tissue culture technique for plant propagation in which tissue is taken from a plant with desirable traits and cultivated in a laboratory to produce offspring that are identical and also contain the desirable traits of the parent plants. Plants that are grown through tissue culture show a trend of producing yield that grows taller and is more disease resistant. Many developing countries rely on this technique to produce a proper yield that is disease and fungus free. Another plant bioengineering technique that is a form of tissue culture is in vitro selection. This process allows scientists to grow and study cells under microscopic surveillance while the cells are put under unideal, adverse environments such as frost, drought, and salts and metals in the soil. This allows the cells to build up a certain immunity against any factors that could prevent the plant from thriving or producing a proper yield. The technique shown in this image is the process of creating a transgenic crop. Transgenic crops are organisms that have been genetically improved through using the recombinant DNA technique of splicing foreign DNA into the coding area of the genes that are to be modified. These genes are inserted into the DNA of these crops, could be of the same species, or it could be of an unrelated species of plant, as well as another type of organism, such as a plant or animal. This technique of splicing DNA allows the advancement of genetic variability while being able to create crops that are nutritionally fortified. There's a type of rice called golden rice that takes 2 DNA sequences of daffodils and then another sequence of a bacterium that makes the rice fortified with provitamin A. This creates a crop that not only has genes from other crops, but it has a gene from a bacteria. It's pretty awesome. That's a crash course on how GMOs work. If you are feeling worried about the safety of GMO products, remember that they are regulated by the Department of Agriculture, the Environmental Protection Agency, and the Food and Drug Administration. It would not be available for consumer purchase if they were not stabilized. In fact, the FDA regards most GMO foods as presumptively safe and thus not needing pre-market approval, but a GMO product that offers significantly difference in structure, function, or composition from substances currently found in food require pre-market approval as a food additive. Now that you have a better understanding of what genetically modified organisms are, I hope that you can feel safe, confident, and proud to consume a product that was researched and enhanced to feed you and your family while making your life more convenient. Last but not least, don't forget to thank the farmers and the scientists that make sure you have clothes and food to eat. So remember, GMOs don't look like a scary monster. They look just like this.
Well, it's 1 for the money, 2 for the show, 3 to get ready. Now go, cat, go, but don't you step on my blue suede shoe.